Hello everyone, I Pratibha Sharma, PGT Biology from Kendriya Vidyalaya, Junagadh. Today under chapter number 2 of class 12, we are going to study following topics. First is outbreeding devices, then pollen-pistil interaction, artificial hybridization and double fertilization. In my earlier class, we studied about the pollination, what are the different type of pollination and what are the adaptations seen in flowers for a particular kind of pollination. So let's start with outbreeding devices. So we find that in majority of flowering plants, they produce bisexual flowers having both pistil and stamen in the same flower. So there are chances that the pollen grains of the same flower, it might pollinate the pistil. Okay, so this type of pollination where the pollens of the same flower come in contact with the stigma of the pistil is known as self-pollination. If self-pollination occurs for several generations, it leads to inbreeding depression. Means it reduces the productivity and fertility of the plant. So in order to avoid this, the plants, they must show cross-pollination. So there are mechanisms by which the self-pollination is discouraged and cross-pollination is encouraged, means which promotes cross-pollination. So such mechanisms or devices are termed as outbreeding devices. They prevent self-pollination and promote cross-pollination. So what are the different mechanisms that prevent self-pollination and promotes cross-pollination? In that, okay, and what advantage we are going to get through it? First advantage is because of cross-pollination, this will help in preventing inbreeding depression. So the fertility and productivity of the plant can be maintained. Second, it results in production of genetic variants, which will enable the plant to survive in different diverse conditions. So first outbreeding device is unisexuality. Unisexuality means a plant produces unisexual flowers. So, if a plant is producing unisexual flower, cross-pollination is the rule, means in self-pollination will not occur. It is seen in dioecious as well as in monoecious plants. So, in nature, we find three types of plants. A plant which bears bisexual flower. Okay. Second, so such plants which bear bisexual flower and the plant where the flowers are unisexual but both the kind of flowers, pistillate and staminate, they are present on the same plant. They are termed as monoecious plants. And then there are dioecious plants where the pistillate flowers are present on a separate plant and staminate flowers are present on a separate plant. So there are two forms. One will be male plant and other will be female plant. Example, watermelon and papaya. Okay, so in this case, the first phase, self-pollination can't take place. Okay, and if the flowers are chasmogamous, then cross-pollination can also occur. But in case of monoecious plant, we will find that autogamy will not occur since the flowers are unisexual. But gitnogamy may occur. Example, cucumber and maize plant. Whereas in dioecious plants, both autogamy and gitnogamy are not possible. So only option is cross-pollination. So in this way, when a plant produces unisexual flowers, then autogamy is prevented and it encourages cross-pollination. Second outbreeding device is dichogamy. This is seen in case of bisexual flowers. Here the stamens and pistil, they mature at different times. There is no synchrony between the release of pollen grain and receptivity of stigma. In some plants, we find that the male reproductive part, it matures earlier than the female reproductive part, that is pistil. This condition is known as protandry. So, when in young flowers, the stamen matures and anther releases the pollen grain. When pollens are released, the stigma of the same flower is not receptive. So, these pollen grains will pollinate the older flowers where the pistil becomes receptive, but it has lost all its pollen grains. So, this is known as protandry, where stamens mature earlier than the stigma. Second is protogyny. In protogyny, when the flowers are young, stigma becomes receptive, but at that particular moment, the anther, they are not dehisced and their pollens are immature. Now, as the flower grows older, the stigma loses its receptivity and the pollen grains are released from the anther. So, this condition where the female reproductive part matures earlier than the male reproductive part is known as protogyny. Okay, so here the young flowers are pollinated by the pollens from the older flower. So, in this way, due to difference in maturation of male and female reproductive part, the self-pollination doesn't occur in these flowers and cross-pollination is the rule. Here you can see protandry condition. So, pollen released on day 1. Day 1, pollens are matured and they are released from the anther, and stigma becomes receptive on day 2. At that time, all the pollens are lost. Okay, so this stigma will receive pollen from some other young flowers whose anthers are mature. In protogyny, here you can see on day 1, the stigma becomes receptive, and then on day 2, stigma loses its receptivity. At that time, pollens are released. Okay, so this way cross-pollination is ensured in some of the plants producing bisexual flowers. Next is heterostyle. Some species of plants, they bear 2 to 3 types of flowers, and these flowers, they differ in the length of their style. So, you can see here in this particular flower, the style is long, and in the second one, the style is short. So, the position of anther and stigma is such that the pollens of the same flower, they can't pollinate the stigma. Okay, due to their different positions. So, pollination thus occurs between the flowers having same length of style and stamen. So, pollination, here the pollen of this particular flower will pollinate the long style flower, and the short style flower, it will be pollinated by the stamens of long styled flower. So, this is another mechanism for ensuring cross-pollination. Here you can see primula. It also shows heterostyle. This one is the flower with short style, and this one is the flower having long style. Next mechanism is self-sterility or self-incompatibility. Here, the pollen grains of the same flower or different flower present on the same plant, if it is deposited over the stigma, then the pollen grain will either not germinate, or if germinates, the growth of pollen tube will not occur. So, self-pollens, they are not able to fertilize the egg present inside the ovule. So this mechanism is known as self-sterility or self-incompatibility. It is a genetic mechanism and is controlled by an S gene. This S gene which is there, it has several alleles. Now the flowers which are present on the same plant will have similar alleles. So for example, you can see here the pistil is having two alleles, S1 and S2. Now the pollen grains of the same plant, okay, pollen grains of the same plant, when they are produced from the microspore mother cell, 50% of them will have S1 allele and other 50% will have S2 allele. And then they come in contact with the stigma. Due to these similar alleles, pistil is also having S1 and S2 and the pollens are also having S1 and S2. So these pollen grains, they will either not germinate, and if at all they germinate, their pollen tube will not be able to reach the ovule. And the egg present inside the ovule. Now, if pollen grain from some other plant, okay, if it falls on the stigma, then in that condition, you will notice that, for example, this one, so they will have different alleles like S3, S4, and here in the pistil, S1, S2 allele is there. So both the pollen grains will be able to germinate on the stigma as they are right type of pollen grain, and they will reach the ovules, and inside the ovule they will fertilize the egg. In this case, you can see second one, the pollens which are arrived from different plant, it is having S1 and S3 allele. Pistil is having S1 and S2. So similar type of alleles, that pollen will not be able to germinate, but the other one will be able to germinate. So in this way, they ensure cross-pollination and prevent the fertilization of egg by the pollens of the same plant. It is seen in tobacco, potato and crucifers. Same example is shown over here. The plant is having S1, S2 allele. So the anthers will produce pollen grain containing S1 and S2 allele. Okay, so both these alleles are there in the pistil. So these pollen grains of the same flower will not be able to germinate. And if they are germinating, their pollen tube will not grow. Okay, whereas S3 is not common, so the pollen grain will germinate and will be able to fertilize the egg present inside the ovule. Our next topic is pollen-pistil interaction. Pollen-pistil interaction, it involves all the events which occur from the time of deposition of pollen grain on the stigma till the pollen tube enters inside the ovule. So all such events are together termed as pollen-pistil interaction. Now when pollination occurs, it does not guarantee that the transfer of right type of pollen grain will occur on the stigma. Sometimes pollen of some other species or of the same plant, if the plant is self-incompatible, it also lands on the stigma. So in that case, the pistil has the ability to recognize whether the pollen received is right type, that is compatible, or wrong type, incompatible. If the pollination is compatible, then the pistil will promote the germination of pollen grain on the stigma and further the growth of pollen tube, which will reaches the ovule. But if the pollination is incompatible, okay, or wrong type of pollen grain lands on the stigma, so the pistil will reject the pollen grain and will inhibit the germination of pollen grain on the stigma or the growth of pollen tube through the style. So compatible pollination, in that case the pollen grain germinates on the stigma and its entine grows out through one of the germ pore of exine and forms the pollen tube. All the content of the pollen grain, it flows down into the pollen tube. Now, if the pollen grain is shed at two cell stage, that is vegetative and generative cell. In that case, the generative cell, it divides and forms the two male gametes in the pollen tube. But if the pollen grain is shed at three cell stage, in which one vegetative cell and two male gametes already present, in that case, the male gametes will be carried by the pollen tube right from the beginning. The pollen tube travels through the style and then enters into the ovule through the micropyle. After entering through the ovule, it enters into one of the synergid through the filiform apparatus of that synergid cell. So all these events, starting from pollen deposition to the entry of pollen tube inside the ovule, are together termed as pollen-pistil interaction. Next topic is artificial hybridization. You can also call this as artificial cross-pollination, that is the pollination carried out by we humans. It's a method it is an approach used for crop improvement programs. Our next topic is artificial hybridization. This can also be said as artificial cross-pollination carried out by agriculturists to obtain the desired hybrid. Okay, so artificial hybridization is one of the major approach of crop improvement program. It involves pollination of desired female plant with pollen from the desired male plant. This is carried out by taking all the precautions to prevent contamination of stigma with unwanted pollen grain. The hybrids which are obtained, they have the characteristic of both the parents and they are superior to both the parents chosen for the cross. Now, if the female plant bear bisexual flower, so artificial hybridization involves two processes or two steps. First is emasculation and second is bagging. Coming to the different steps of artificial hybridization, the first step is selection of parents for the hybridization. The parents are selected, male and female parent. They are selected on the basis of the objectives of the cross. Suppose we want the hybrid should give higher yield as well as it should have environment resistance to environmental stresses. So we select two parents having these characteristics in order to obtain a superior quality of hybrid. So first step is selection of parents. After that, next step is emasculation. If the flowers produced by the plant are bisexual, then in that case, emasculation is performed on female flowers where when the flower is in bud condition or when the anthers are not dehisced in immature state, in that case, we remove all their anthers with the help of forceps. Okay, so this process of removal of anthers from the female flower is termed as emasculation. But suppose if the flowers are unisexual, then emasculation is not required to be conducted on female flower. Emasculation prevents self-pollination. Next step is bagging. Once the flowers are emasculated, then they are covered with a polythene bag of suitable size. It might be made up of polythene or butter paper. By bagging, we prevent the contamination of the stigma with unwanted pollen grain. After bagging, the next technique followed is known as tagging. The emasculated flowers are tagged just after the bagging. The tags are attached at the base of the flower or inflorescence with the help of a thread. The tag contains following information. that is the date on which the emasculation is carried out, the date on which the pollination is carried out, and the name of male and female parents. The name of female parent is written first and then the male parent. Once the stigma is receptive, pollen grains are collected from the male parent. The bag of the female flower is removed and pollen grains are dusted over the stigma of emasculated flowers. And the flowers are again rebagged until the fruit develops. So all these steps are carried out in artificial hybridization. Whereas if the female parent bears unisexual flower, so we directly starts from bagging, tagging, and rest of the steps will be similar. By this technique, we obtain hybrid which is superior to both the parents. Now the next topic is double fertilization. This is the picture of embryo sac which is formed inside the megasporangium, that is ovule. Embryo sac is also known as female gametophyte. So double fertilization is a complex process which involves fusion of one female gametophyte, that is one embryo sac fuses with two male gametes. This double fertilization is the characteristic of angiosperms. It involves two fusion, one is syngamy and the second one is triple fusion. So as we studied in pollen-pistil interaction, as the pollen grain lands over the stigma, due to pollen-pistil interaction, the pollen is recognized as right type or wrong type. It germinates and it forms the pollen tube carrying two male gametes. The pollen tube then enters inside the ovule through the micropyle and then enters into one of the synergid guided by the filiform apparatus of that synergid. Here, after entering into the synergid, the pollen tube's tip, it bursts and it releases the two male gametes. One of the male gamete immediately fuses with the egg cell present in the egg apparatus and results in the formation of diploid zygote. This zygote later on develops into embryo. The second male gamete then passes into the central cell and fuses with the two polar nuclei. This fusion is termed as triple fusion since it involves fusion of three nuclei. After fusion, it forms primary endosperm nucleus and the central cell gets converted into primary endosperm cell. This primary endosperm cell later on develops into triploid endosperm, which provides nourishment to the developing embryo. This is the structure of fertilized embryo sac which is asked in exams. So you have to practice. If it is like you might be asked to draw the structure of embryo sac, then the previous one before fertilization, the structure which is there, that you have to draw. But if it is asked that draw the diagram of fertilized embryo sac, so you need to draw this diagram. This here you can see central cell got converted into primary endosperm cell and this three nuclei, they fused and formed the primary endosperm nucleus. So these two structures are the result of triple fusion and the zygote is formed due to syngamy between male and female gamete. Okay, so this is how the double fertilization is taking place. Now here there are few board questions. Go through these questions and if any query is there, you can ask me. Thank you.